For 500 years, one group of believers built their own world across 4 continents. Dry land in Ukraine and wild land in South America became some of the richest farmland of their time. Schools, aid funds, and medical clinics followed, paid for through community auctions without asking any outside government for a thing. They even built their own language, called Plautdietsch, spoken nowhere else on earth, and for 5 centuries they married almost only within their own faith. You've probably heard the simple version of this story. Buggies, handmade clothes, a quiet life cut off from modern science. That picture comes from something real, but it hides an assumption underneath that a community this closed off must be helpless against its own biology, along for whatever their genes decided. What they actually did with that discipline reached all the way into their bodies. Every marriage inside the group left a mark scientists can still read today in their DNA. There is a specific reason for it. No single person alive chose it on purpose, but every one of them helped write it into the next generation. But what their DNA actually reveals is more disturbing than the popular picture. It's not what you'd expect. By the end of this video, you will never think about genetic isolation the same way again. Before we start, if you follow stories where the science changes what you thought you knew, subscribe now. This is one of those stories. Mainstream media loves one version of Mennonite life. Reality shows treat the community like a strange leftover from the past. That picture hides an idea. It suggests staying apart from the modern world means staying apart from modern science too. It also suggests these communities are passive victims of their own biology. Real, peer-reviewed science tells a different story. Peer-reviewed means other scientists checked the work before it was published. In 2017, a team led by Howe read the full genetic code, also called the genome, of 265 Anabaptist people. Reading someone's full code like this is called sequencing. That project happened because plain communities took part as full partners, not studied from a distance. Together, they built some of the most advanced community health programs in the world. It is true that marrying within the group over and over across 5 centuries raises the risk of certain inherited diseases, and that much of the popular idea is correct. But the response to that risk has been active and organized. That same community built real, working institutions long before any scientist arrived. They ran their own aid system, called the Waisenamt. It paid for medical care and financial support for families in need, all without any outside government. Here is the number that stops you cold: 14%. That is all the genetic variety Anabaptist communities carry. Compare that to a typical outbred European population, which carries the full range. Outbred means a population where people marry widely, not only within one small group. That 14% number comes from Cheda, Hu, and their team, published in 2023. 500 years of marrying within the group did more than shape a lifestyle. It filtered an entire genetic code down to a small slice of what it once was. But paper records can only tell us so much. To see what that isolation really did, we need to look at the DNA itself. In 2016, a team led by Pardo Seco published a study in the journal Scientific Reports. It was called Genomic Continuity of Argentinian Mennonites. The team set out to map one settlement, called La Nueva Esperanza, in Argentina. Their data revealed something bigger: a full 500-year migration story. This settlement's ancestors started in Switzerland and the Netherlands in the 1500s. Persecution pushed them to the Vistula Delta in Prussia. Later, Catherine the Great invited them into the Russian Empire. They built 2 colonies there, Chortitza and Molochna. New laws in the 1870s threatened their exemption from military service. Thousands moved to Manitoba, Canada. Schooling disputes in the 1920s pushed another group to Chihuahua and Durango, Mexico. Finally, in 1985, one group settled in La Nueva Esperanza, Argentina. The research team studied more than 580,000 genetic markers. These are small, specific spots in a person's DNA that scientists can compare. This group had lived in South America for decades, surrounded by people with mixed European, Native American, and African ancestry. But the Mennonite genetic code showed 0% mixing with any of those outside groups. The closest genetic match was a European reference group from Utah, used in a major worldwide DNA project called the 1000 Genomes Project. The father's side DNA traced straight back to North German and Dutch founders, too, and the study held up well under close review. That same large sample, a clear founding population, one consistent result no matter the DNA test used. A second team, also led by WHO, published a related study in Scientific Reports in 2017. They sequenced those same 265 Anabaptist people. That project found more than 12 million genetic differences, called variants. Of those, 12.5% were brand new, unique to this population's own history. That number is a clue Section 3 will explain. Mennonite communities carry a genetic signature found nowhere else on Earth. It was written 5 centuries ago, and it is still changing today. But the big picture numbers can only tell us so much. The most important discovery came when researchers looked closer. They wanted to know what isolation did, generation by generation, to the genetic code itself. Here is how this works, in plain terms. Picture copying one book by hand over and over for 500 years, only ever copying the copy, never checking it against any outside library. Small mistakes either disappear or spread into every future copy, and nothing new ever comes in from outside. That is close to what happens inside a closed group that only marries within itself. A small starting set of genes passed down, generation after generation, with no new genes coming in. Rare genes that would normally stay rare in a large open population instead spread. Copy by copy, they become common. Scientists call this a founder effect. It is also how brand new gene variants, like the 12.5% we mentioned earlier, end up unique to one population, copying mistakes with nowhere else to go but forward. 6 named teams have documented these diseases running at rates far above normal here. Strauss and his team, working with Dr. Morton's team, studied glutaric acidemia type 1, caused by a mutation, a change, in a gene called GCDH. This disease strikes about 1 in 500 Mennonite babies. Compare that to the general population, where it strikes about 1 in 100,000. Puffenberger and Morton's teams studied maple syrup urine disease, caused by a change in the BCKDHA gene. Among Old Order Mennonites, about 1 in 7 people carry this gene change. Up to 1 in 178 babies are born with the disease. In the general population, that number is closer to 1 in 185,000. Castrinos and his team studied congenital nephrotic syndrome, caused by a change in the NPHS1 gene. It strikes about 1 in 500 Mennonite babies, roughly 20 times the already high rate seen in Finland. Greenberg and his team found that in Manitoba's Old Colony, Mennonite communities, 1 in 25 people carry a gene change linked to hypophosphatasia, a disease that weakens bones. 2 more diseases round out the picture. They show how narrow this compression can get. Cruz Aguilar and his team studied Mennonite communities in Durango, Mexico. There, almost every documented case of Alström syndrome, a serious disease affecting vision, hearing, and the heart, traces back to one gene change in a gene called ALMS1. In the general population, Alström syndrome affects fewer than 1 in 1 million people, and Strauss and Puffenberger, studying the Grofdale and Weaverland communities, found that almost every documented case of megaloblastic anemia traces back to a single change in the AMN gene. This disease is so rare elsewhere that most doctors will never see a single case in their whole career. Genetic testing shows how far this pattern spreads. Among married couples here, 24.3% are both carriers of the exact same harmful gene, meaning they carry one copy, which usually does not make them sick. And 5.2% carry 2 copies of a known harmful gene variant, one from each parent, which is what usually causes disease. The big picture numbers told one story. The generation by generation story told something else. This is more than a snapshot of Mennonite genetics today. It is a record that keeps growing, one generation at a time. It tracks every family that stayed inside the line. Think about what that means. The mother's side story matches this pattern exactly. In 2010, Melton and his team studied mitochondrial DNA. This is a special kind of DNA passed only from mother to child. They studied 6 Mennonite communities in Kansas and Nebraska. They found 58 different maternal gene patterns. Only one of those 58 was shared across all 6 communities. In the Old Order Mennonites of Garden City, Kansas, one single maternal pattern made up as much as 75% of the entire maternal line. Other isolated groups show this same pattern too. The Amish, the Hutterites, certain island populations, they all show similar drops in genetic variety. What makes the Mennonite story worth telling on its own comes down to scale and time. This is 4 separate narrowing events, not one. Each is fully documented. Together, they cover the same 4 centuries and 3 continents you heard about a moment ago. Switzerland and the Netherlands, then Prussia, then the Russian Empire, then the Americas. At every stop, whole families and church groups moved together. Researchers Crawford and Melton call this kin-structured movement. Families moved as units, not individuals. And at every stop, even after living for decades near very different populations, the genetic code came out the other side with 0% outside mixing. This is isolation that survived 4 moves, 3 continents, and 500 years, fully intact. The same community system that made all of this possible was also doing something else, something written into every cell of every person who stayed. Menno Simons lived this story. His life is the closest thing we have to a living record of how it all began. Born in Friesland in 1496, he trained as a Catholic priest. In 1536, with believers being hunted down and killed across the region, Simons joined them anyway. He pulled the movement's scattered hunted pieces into one tradition, built on adult baptism, chosen by each believer rather than assigned to a baby at birth, and on strict discipline for anyone who stayed. That discipline, not any government or border, kept the gene pool sealed for 5 centuries. It is still traceable today. The Argentina study from Section 2, the one that found 0% outside mixing, traces straight back to the line Simons drew in the 1530s. Then, there is Dr. D. Holmes Morton. In 1989, he traveled to Lancaster County, Pennsylvania. He was a children's doctor and geneticist from the Children's Hospital of Philadelphia. He found Amish and Mennonite babies with unexplained brain damage. At the time, locals called it Amish cerebral palsy. Dr. Morton found the real cause. It was glutaric acidemia type 1, the same disease driven by that GCDH gene change, striking about 1 in 500 Mennonite births. Instead of sending families to distant, expensive hospitals, Dr. Morton and his wife, Caroline, built the clinic for special children on donated land in Strasburg, Pennsylvania, with volunteer labor from the Plain community itself. Before the clinic existed, more than 95% of children with this disease suffered permanent brain damage during ordinary childhood fevers. With same-day testing and fast treatment developed right at the clinic, that number dropped below 5%. The clinic also pioneered a liver transplant cure for maple syrup urine disease. It has performed more than 100 successful transplants. Altogether, the clinic has saved an estimated $270 million in medical costs families would have otherwise paid at distant hospitals. Modern genetic testing has confirmed the exact same gene changes Dr. Morton chased in the 1990s, still traceable today in the very same families, generations later. They built something that has lasted 500 years. But the same system that protected that world was also shaping something else, cell by cell, in every person who honored it. Individual stories can only take us so far in showing how. Only 14% of typical outbred variety, 0% outside mixing across 4 moves and 3 continents. That compression has a cause, and it is still shaping Mennonite DNA worldwide today. The cause has a name and a date. In 1527, Michael Sattler wrote the Schleitheim Confession, a statement of religious belief and rules. Article 2 set up the Ban, or Meidung, cutting off any member who left the faith. Article 4 called for strict separation from outsiders. Those rules became daily practice through the Ordnung, the community's unwritten rulebook, allowing marriage only between baptized church members. The numbers behind this system are real, too. Studies show 80 to 90% or more of young people finish Rumspringa, the teen decision period, choosing baptism and staying. That high rate keeps outside genes from entering the population year after year. Crawford in 1989 and Melton in 2010 showed how that closed loop grew over time. When a colony grew too large, whole family groups moved together to start new ones, rather than splitting at random, a pattern called kin-structured fission-fusion. Each split created a new, smaller founding group and concentrated the same rare genes all over again. Think about what that means. One man wrote a statement of faith in 1527. No government stood behind it, no law enforced it, yet it worked for 500 years, the way an outside legal system might have. Community members were the ones who kept choosing this system, generation after generation, at Rumspringa, at marriage, at every new colony founded along family lines. And still, this system shaped the genetic identity of every person who honored it, one generation at a time. Descendants who take a DNA test today find the personal present-day face of that same mechanism right now. We will show you exactly what that looks like next. The genetic record of 500 years of choices is still there, and it cannot be undone. What the DNA kept and what it is telling researchers and descendants today is what we cover next. The Ordnung could control who married whom. The ban could enforce community lines through shunning. 4 moves across 3 continents could scatter the population across half the globe, but none of it could reach inside the genetic code or undo what 5 centuries had already written there. The DNA kept its own record. Commercial and clinical DNA tests are now bringing that record to light. Descendants who take a genetic test often find long matching stretches of DNA shared with people who look distant or unrelated on paper. In a population this tightly linked, normal labels like second cousin start to break down. That same tight linkage has become useful far beyond the community. The Anabaptist Genome Reference Panel, built from those 265 genomes, now supports research into mental health, body chemistry, and heart disease worldwide. A population that isolated itself for reasons that had nothing to do with science has become one of the more useful genetic records on the planet. That relationship with science has a thoughtful side, too. Plain communities take an active part in screening and testing for diseases that can be treated, the same testing that let Dr. Morton's clinic cut GA1 brain injury from over 95% down to under 5%. But they show more caution around tests for untreatable adult-onset conditions, where a result could only change how many years a person spends knowing about it, not the outcome itself. It is the same practical family-first thinking that built the clinic for special children. Get involved where science changes what happens next, and hold back where it would not. Follow the thread back to the start. The myth said isolation meant standing still. The genetic cluster said otherwise, a population with a clear traceable identity all its own. The core twist said isolation adds up year after year, growing whatever genes were there at the start. Menno Simons and Dr. Morton showed what real people built and protected inside it. The Schleitheim Confession and the Ordnung showed exactly how, without a single government document. And the DNA, still readable today, shows none of it could be undone. What the DNA revealed about Mennonite communities is disturbing, precisely because it is so human. 500 years of choices, each one made by a real person honoring a real promise. They added up in the genetic code, generation after generation. They built something that outlasted the empires around them. Their DNA recorded every year of it. That is a living record of what it means to choose your world and to stay inside that choice for 500 years. Picture them one last time, a small group of families moving from Switzerland to Prussia, to Ukraine, to Canada, to Mexico, to Argentina. They carried the same faith and the same discipline. And, without ever meaning to, They carried the same DNA all the way to today. If this changed how you think about genetic isolation, about community, about what 500 years of choices can write into a body, subscribe. Share it with someone who thinks they already understand what isolation does. And tell us in the comments, which community's genetic story should we investigate next?